Yeah, welcome to the morning session here, beginning of the Analytica conference, and I'm pleased here to introduce to you the session. Our session is about analyzing emerging hazard in foods, and well, it's a session mainly organized and dedicated for food chemists and, of course, also to other disciplines. And first of all, I would like to announce that for each da muss ich auf Deutsch das reden, weil sonst kann ich die Spezialworte kenne ich nicht. Es gibt eine ZFL-Zertifizierung für Lebensmittelchemiker. Das heißt, man kann für diese Session und für die Session heute Nachmittag zu FOODOMICS kann man Zertifizierungspunkte erhalten. Und wenn Sie solche Zertifizierungsfortbildungspunkte haben möchten, dann tragen Sie sich bitte auf dieser Liste ein. Die liegt hier dann aus und dann können Sie dann von der GDCH dann diese Punkte erhalten. Okay, so that's all for the Germans. And well, let's start. So emerging hazard, and it's a particular pleasure to introduce our first speaker to you. It's Sarah de Saeger, and I think we know for quite a long time, had some yes, interesting engagement, and as well we met on several journeys, interesting journeys as well, particularly, and I would like to introduce her as well. She's the head of the Center of Excellence in Mycotoxicology and Public Health, and full professor at the University of Ghent, and as well. Her specific, let's say, engagement is in the South Hemisphere, I would say, and particularly in Africa. And she's the coordinator of the international thematic network MYCOTOX-SOUTH. Means she is particularly engaged in Africa, I would say sub-Saharan Africa. That's your focus, yeah. And in that view, she is visiting professor at the University of Johannesburg since 2020. She's as well president of the International Society of Mycotoxicology. Has been elected in 2021. And as well, which I think is really prestigious, is member of the Royal Flemish Academy of Belgium for Science and the Arts. Well, and I think, well, we have been engaged in many occasions on the mycotoxins, and that's as well what she will be talking about. So the topic of her talk is recent developments in the bioanalysis of mycotoxins and their modified forms. So, Sarah, the floor is yours. I hope that will work. Right.
Thank you very much, Michael, for this very nice introduction. And it's very nice for me to be here in Munich. It's my first time at the Analytica conference, so I'm looking really forward to the next days. Okay, but I don't know this. Okay, yeah. So, as Michael said, indeed, I will talk about recent developments in analysis of mycotoxins and their modified forms. I will start with a brief introduction on food security and mycotoxins. Then I will mainly discuss with you the different mycotoxin analytical methods. Afterwards, I will go towards the human biomonitoring and exposomics. And finally, I will say something very short about what has been introduced on my collaborations in the South and the MYTOX-SOUTH network. So, of course, we all know that food security is nowadays under pressure because of many different issues. First of all, there is, of course, the global population growth. There is climate change. There are conflicts and wars all over the world. And, of course, there is also the coronavirus. So all of these have indeed made it much more difficult to reach the Sustainable Development Goal number 2, which is on zero hunger. So if you look to this definition of the SDG 2, you can see that it's not only about having enough food or having sufficient food, but also having safe food. So indeed, food safety is part of food security. And if we look to food safety on a global level, then indeed mycotoxins are one of those biggest obstacles to reach safe food or to reach the Sustainable Development Goal number 2. So what are mycotoxins? Those are food contaminants that we will find in many different plant-based foods, like cereals, fruits, nuts, spices, but also in animal-derived products like milk, if the cow has been fed with contaminated feed. So those mycotoxins are produced by fungi. Those are secondary metabolites, which means that they are not necessarily needed for the growth of the fungi, but they have pharmacological or toxic effects. And we all know that some very important medicines have been derived from fungi. For instance, penicillin, which is an antibacterial, derived from Penicillium and Aspergillus species. Also, lovastatin as an example, which is an anti-cholesterol agent derived from Monascus purpureus. But most of those secondary metabolites have a too much acute toxicity and cannot be used as medicinal drugs, but are indeed toxins. The most famous here is aflatoxin B1, which is the most potent mycotoxin because it's a liver carcinogen. Now, aflatoxin B1 is the most famous, but it's not the only mycotoxin. There are many, many mycotoxins, hundreds are known, and here you can just see a list of the most important ones, like ochratoxin A, patulin, deoxynivalenol, zearalenone, fumonisins, ergot alkaloids, Alternaria toxins. They are all produced by different molds from different genera, like Aspergillus, Penicillium, Fusarium, Claviceps, and Alternaria species. So this brings me now to the analytical methods, because it's very important that we are able to control them in our food and feed. Now, there are some general challenges in mycotoxin analysis. First of all, as you have seen on this slide, they have very different chemical structures, very complex chemical structures. They are all small metabolites. Another challenge is that they are present in very low levels, so in the ppb and ppt range of concentrations. Also, they are co-occurring, so mostly in one food matrix you will find different types of mycotoxins. So therefore, the analytical methods should be able to analyze different mycotoxins at once. Also, the matrices, the food matrices, are very complex. And besides that, mycotoxins are not only present in their parent form, but can also be present in modified forms. And this is what I want to show you here. So, depending on the origin, mycotoxins, the chemical structure, can be modified. For instance, the fungi themselves already can produce different derivatives of mycotoxins. For instance, deoxynivalenol can also be in the form of acetyl deoxynivalenols produced by the fungi. For the plants, when they are infected with the fungi, they will start up like a defense mechanism against the toxin, and they will bind the toxin to glucose to have it conjugated and less toxic for the plant. So then we have, for instance, deoxynivalenol 3-glucoside. Also, food processing can result in different modified forms. Here it's also important to tell you that mycotoxins are very stable molecules, so just heating will not reduce the level, but it can result in different forms. Then, in animal and human, we have metabolization of mycotoxins to different phase 1 and phase 2 metabolites. And for instance, a well-known example of a phase 2 metabolite is deoxynivalenol 3-glucuronide, which we can measure in urine. Also, if we look to the analysis of mycotoxins, in fact, the sampling is the most important step, and sometimes this is forgotten, and we need really a representative sample because this is the largest source of error in the analytical process. And this is because mycotoxins are present in hotspots. The fungi are present at several spots in a big lot, and therefore it's important that for the sampling you collect different incremental samples in one lot and then put them together in one aggregate sample. So concerning this, there are many different guidelines in the European Commission legislation. So if we then have a look to the different existing analytical methods, we can make a difference between screening methods and confirmatory methods. Screening, as you all know, is just to know, is the mycotoxin present at a certain level, yes or no? So here, the typical methods are same as for many other different food contaminants. We have ELISA, we have fluorescent labeled immunosorbent assay, we have fluorescent polarization immunoassay, chemiluminescence immunoassay, different biosensors, and then of course for real qualitative results we have lateral flow or dipstick tests. A little bit more on the ELISA, which you probably all know. There are a lot of advantages. It gives you some quantitative results. It's quite fast technique. You don't need much sample. And for mycotoxins, very important, they are present commercially for all mycotoxins in many different food matrices. So this is a well-covered market. Concerning modified mycotoxins, however, it depends on the antibody which is used in the ELISA if you will be able to detect a modified form, yes or no. Of course, there are also limitations, and I think the main limitation is that you can only use it in the lab. You can't use it, for instance, on the field. Now, if you want to make your ELISA more sensitive, for instance, for toxins like aflatoxin M1, which are present in milk at really very, very low concentrations, then you can change your label to a fluorescent label, for instance, to a quantum dot, and then you can increase the sensitivity of your method. Concerning the limitations, similar as for ELISA. So it can also not be performed on site. If you really want to have a field test, then you need to work with lateral flow immunoassays or dipsticks. We all know them now with the COVID testing, of course. And those are very low cost, fast, can be used, for instance, in developing countries for a very quick screening of a sample. And for the future, I think this is the way to go for further biomarker diagnosis in human. I will go on that further on that aspect of the analysis of mycotoxins in human biofluids. Of course, there are many limitations. You can only use them for certain not too complicated matrices because the membrane surface is very prone to interactions and matrix effects. So this brings me to the confirmatory methods. And here, I would say the technology that most of mycotoxin researchers are using nowadays is LC-HRMS, so liquid chromatography, tandem mass spectrometry, because then you can have a multi-mycotoxin method. You can also use it for any kind of sample, food, feed, but also for the human biofluids. You can use it for urine, serum, plasma, feces, and so on. Generally, you start with a sample preparation. This can be grinding of the cereals, homogenization. Then you have the analysis with an extraction, followed mostly by a sample cleanup to have it more specific and to get lower LODs and LOQs, and then detected in the purified extract. So with LC-HRMS, we know we can identify the toxin, we can quantify and confirm the presence. Nowadays, there is also more and more a trend toward high-resolution mass spectrometry, LC-HRMS. Here you can work in targeted mode or in untargeted or non-targeted data analysis, and we make use of this technology to discover unknown secondary fungal metabolites, to study the degradation products and also the modified forms, which are, as I said, the result of processing techniques, for instance. And also nowadays we use it to unravel the human metabolism. I will just give you one example of a very recent research which was performed in my lab on the analysis of Alternaria mycotoxins in apples, for which we used both a targeted and an untargeted approach. So this is the work of my recently promoted PhD student Agustina Pavicic. She worked on apples from Argentina, and she used a very easy catchers-based extraction and cleanup, and then she was able to analyze more than 11 Alternaria mycotoxins, including modified forms such as sulfates and glucosides. So as you can see, she made use of a Xivo TQS mass spectrometer, and yes, she also made use of matrix match calibration, as well as some internal standards like the C13 thiazonic acid. For the untargeted approach, she made use of a SYNAPT instrument, and the extraction here was more general, so no cleanup, just extraction with methanol water acetic acid, an evaporation step and a resuspension step, some filtering, and then she put it for untargeted analysis, both in ESI plus and ESI minus approach. Here you can see that she used the Progenesis software to analyze all the data of the untargeted analysis. And at the end, she gets like 21,000 compounds from this experiment. And of course, then the most difficult part starts, is the analysis of all those features that are derived from this analysis. So finally, she could bring it towards like 9,000 compounds, and you can see then she used the PCA, the principal component analysis, to make a discrimination between infected and non-infected. And you can really see, so here were all the infected apples, and the non-infected were clearly discriminated from the infected. Now, of course, we need to find out all those different metabolites, what they are, and the ones which are really related to mycotoxins, to Alternaria mycotoxins. So this work is still ongoing, not yet published, but I hope we will have some results soon. So this was mainly on the analysis of mycotoxins and food and feed. But as I said, in the last couple of years, there is much interest into analyzing mycotoxins in human biofluids to detect the exposure or the direct exposure of mycotoxins through biomarkers of exposure. So also for this, we have now developed two different methods which we can use in plasma, in serum, and in urine. So we have a targeted LC-MS analytical method for 42 mycotoxins and the metabolites. This is with the Xivo TQ-XS instrument. And we have an untargeted approach where we have developed a database for more than 150 mycotoxins and the metabolites that we currently know. So those two methods are not yet published, but we use them now in different studies. A first important type of studies that we are performing are intervention trials, which is part of an ERC starting grant, which was received by my colleague, Professor de Bouvre. So we are co-leading the lab, and she received this ERC, and now she's working on human intervention trials to really study the toxicokinetic of mycotoxins in the human body. So for this, some volunteers, they receive small, very small amount of mycotoxin, which is below the tolerable daily intake. Of course, we cannot do this with the real carcinogenic mycotoxins like aflatoxin. We cannot do it. So we only do it with the chronically toxic mycotoxins. And then through collection of urine, feces, and blood, we really study the toxicokinetics over time. So we do this, based on the publication that you can see, where we already found, for instance, that deoxynivalenol is excreted within 24 hours. So now we are doing it for many different mycotoxins, among others, some Alternaria toxins. So, of course, as I said, we follow over time, so we use a targeted approach to follow one mycotoxin at once, but also here we make use of the untargeted approach. Why are we doing that? Because, still a lot is not known about which mycotoxins or how mycotoxins are metabolized. So this, for instance, is the example of teniazonic acid, which was given to the volunteers, and then we analyzed. This is urine. We analyzed the urine of the exposed and the non-exposed groups, and again through PLSDA we could discriminate the two groups. So the exposed ones is the red, and the non-exposed are the blue subjects. So again, here we found, of course, a lot of variables which can be responsible for this discriminating effect. And now, so it's my PhD student Lia Vizintin who is working on that together with Eugenia Aladio from the University of Torino, who is doing the bioinformatics part. So now she could already found out that 11 of those features were real metabolites of teniazonic acid, and we are now further unraveling the structure. Just to show you, as I said, the most difficult part is the bioinformatics part, so we had to look for additional expertise in other labs to work on this part. Another example of where we use our multi-mycotoxin biomonitoring is here a study that we performed in Ethiopia on pregnant women, where we made an association between maternal aflatoxin exposure and the fetal weight gain. The left graph, you can see that, so here we use targeted approach. We just measured the aflatoxins in the human plasma. And you can see, so we made an association between the amounts of aflatoxin in the plasma and the weight gain, the estimated weight gain of the fetus from the pregnant women. And you can see that we saw a significant difference, so that there was a lower fetal growth in the exposed women. So also for this study, we are now looking to an untargeted approach, to a metabolomics approach, to see if there are differences between the high exposed women and the non-exposed women. And here we are also looking if there are some distinctive feature or biomarkers of effect that we could use in the future to make, to better understand exposure of mycotoxins in women. Of course, we all know that mycotoxins are just one of those many compounds that we are exposed to. And therefore, it's important that we all broaden our horizon and that we are not just looking to our own niche of work. So for this reason, we are now trying to go towards the exposomic approach, which means that we are trying to measure cumulatively all the environmental influences that we are exposed to throughout our lifespan. And so this means that we are looking to the external exposome, which you can measure through environmental monitoring, some specific personal exposome through biomonitoring and questionnaires, and then we also look to the biological responses through omics methodologies. Now, in this respect, we just started last year with the Flemish gut exposome project, FLEXIGUT, which is the first large-scale exposomic study that focuses on chronic low-grade gut inflammation. So here we have 2 Flemish cohorts, which we are following, the environage birth, which starts from the pregnant mother until the children, and then the Flemish gut flora project, which follows adults. So at the end, we have a sample size of 400 children, 400 adults, from which we measure the complete exposome. Now, in that exposome project, we also have specific hypotheses. And one of them is that chronic low-dose multiple mycotoxin exposure probably has an influence on the gut microbiome inflammation axis. So for this reason, we do biomonitoring of the mycotoxins. We have a lot of metadata. We also measure the gut, the gut microbiome through metagenomics. We perform metabolomics as well as DNA adductomics, all on the same samples. We associate all this data with the chronic low-grade gut inflammation through inflammatory markers. And finally, we want to make associations with diseases like metabolic disorders and gastrointestinal cancers. So again, here, the analysis of everything, metabolomics, DNA adductomics, all of this, I think we are now experienced in this, all the different labs who are working on that. So the analytical part we have, let's say, under control. But again, the difficult part for us is the integrated data analysis, to bring together the biomonitoring data, the metabolomics data, DNA adductomics data, metagenomics, together with those inflammatory markers. So this is our huge challenge on which we are working now. And so this brings me to the end of my presentation, and I just would like to show you something on my real passion, the MYTOX-SOUTH network. Nowadays there is no presentation anymore in which I'm not talking about MYTOX-SOUTH, so just give me 3 minutes. So what is MYTOX-SOUTH? As you can see, it's a global network on capacity building, because we have now all this knowledge on analytical methods, but it's important that this is not just in some few labs throughout the world, but that we build further analytical capacity everywhere in the world where the mycotoxin problem is huge. So here you can see at the moment our current network with our partners. So indeed a lot in sub-Saharan Africa, but nowadays we are also having partners in Latin America and Southeast Asia. You can have a look to the website. There is a very nice video which only takes 3 minutes. So if you want to have a look, you can watch it. So what is our aim? Being from a university, we want to educate and train young students and scientists from low and middle income countries, and we want to build capacity through co-creation. We also work on further awareness creation of the mycotoxin problem, and we are conducting research and developing new technologies for mitigation of the mycotoxin problem. So just a few examples. So we have some money to fund traineeships. Here you can just see a list of people who were already trained in our lab or in other labs of METABOLITES network. For instance, this year we will have 2 people coming to our lab and 1 person will go to a lab in Italy for training. Awareness creation, we do it on different levels, for instance also at the grassroots level through the farmers. So we were having a farmers lab in February 2020, just before the COVID lockdown, in Johannesburg, in Soweto. We also disseminate our results on mycotoxins to policymakers. For instance, these are 2 examples where we were in Nigeria in 2019, together with the Flemish trade mission to Nigeria, but also just a few days ago I was in Nigeria for a 2nd academic mission, where we, for instance, also visited FAO and IFPRI to discuss about collaboration on mycotoxins. This is my final slide. I would like to invite all of you who are interested in mycotoxins to come to the conference, which is organized by the African Society of Mycotoxicology, together with METABOLITES in September in Stellenbosch. So finally, after 2 years, we are also able to organize it. So that's it. Thanks a lot for your attention. If you have any question, you can, of course, ask me.
Yes, thanks. Yes, Sarah, thanks a lot for presenting your vision on the future of our field of the mycotoxins, as well. What I found quite really interesting and challenging is your approach to screening for the co-occurrence, which is, I think, highly challenging for us here, which we don't have a clue yet. And of course, for presenting your engagement in MYTOX-SOUTH, your network all over the world.